Can you hear okay? Okay, awesome. Good morning, guys. How's everybody? Rising on the sun, I know, finally it's the break. I'm really excited to present today. We've been working really hard and it's great to see faces that I recognize and also new faces. So apologies to those that have heard versions of this talk before, but I think it is, I'm very grateful to be here and to have the support of the CIEHS to present this exciting work that we've been doing. So I'm mainly going to focus on talking about our new study that we are developing called Louisville Environmental Assessments Pilot Study in Colorectal Cancer. But I'm going to give some background because by training, I'm a breast cancer epidemiologist focused on environmental exposures. So I've learned a lot about colorectal cancer in this, I guess, 3-year, almost 4-year journey. So just an overview of the talk, I'll go over a little bit about colorectal cancer epidemiology, presenting a lot of statistics from the American Cancer Society. And then I'll talk to you about the work that we did the start of the pandemic on colorectal cancer hotspots in Kentucky. And then I'll introduce our LEAPS in colorectal cancer study and show you all some pilot results for metals exposures, and then talk about our next steps with the LEAPS study. So here's on the top right is a figure of the colon and the rectal or the colorectal. So colorectal cancers can occur anywhere from the cecum all the way through the ascending colon, transverse colon, descending colon, sigmoid colon into the rectum. So it doesn't include anal cancers. Learned that recently. And here we see some of the stages of colorectal cancer. So here is a polyp on the inside, and when it breaks that barrier or that membrane, that's when it becomes a cancer. And when it extends outside of this wall, we would call that a regional cancer. And then when it metastasizes to other organs, that is called a distant colorectal cancer. With cancers like breast cancer and prostate cancer, when we have screening, we generally see a large spike in the number of cases of prostate cancer and breast cancer. The interesting thing about colorectal screening, colorectal cancer screening, is by removing the polyp early, we're actually preventing the development of colorectal cancer. So it's one of these screening tools that actually by screening, we're preventing cancer from developing. So in epidemiologic rates, we don't see this large spike in colorectal cancer incidence with screening. So that's just one interesting thing. And also a reminder, if you're in the screening age, to go get your colonoscopy. Okay, so here's some national cancer statistics for the US. On the top, we have the estimated number of new cancers of the top 10 cancers, and you can in males and females, and you can see that colorectal cancer incidence is the third most common cancer diagnosed, with around 80,000 males being diagnosed each year and around 70,000 females being diagnosed each year with colorectal cancer. On the bottom here, we have the estimated deaths from colorectal cancer, and you can see each year, again, colorectal cancer is the third leading cause of cancer death. Around 29,000 males die each year from colorectal cancer, and around 25,000 females die from colorectal cancer each year. Here we are looking at some statistics from the American Cancer Society. On the far left, we have the colorectal cancer incidence rates from 1998 through 2019. And you can see that generally in all age groups, both localized and regional colorectal cancers have been decreasing over time. So the incidence of colorectal cancer has generally been decreasing over time, thanks to screening and other prevention efforts. However, what's concerning now is this rise in early onset colorectal cancer. So being diagnosed with colorectal cancer before the age of 50 years old. So here in the middle figure, we see the incidence rate of all localized, regional, and distant colorectal cancers increasing over time. Now, I want to also point out the trends in colorectal cancer incidence and mortality by race and ethnicity. While the incidence rates have been decreasing, I want to point out that still the incidence rates of colorectal cancer are highest in Black individuals in the U.S., followed by white individuals and Hispanic individuals, either Black or white, or unpaid. Mortality is also highest in Black individuals in the U.S., and followed by white individuals and Hispanics. So there's been increased public awareness of early onset colorectal cancer. Here's just some screenshots from recent news alerts that I get on my phone. So this is from the Washington Post. Colon cancer is rising in young Americans. It's not clear why. Here's from NBC News and then the NCI. Did anybody go to the U of L basketball game over the weekend? Okay, it was a tough game, but what around one of the TV timeouts, they were promoting. So today is actually the first day of Cancer Prevention Month. It's February. I don't know if they knew that, but they brought out cancer survivors during one of the TV timeouts. And one of those cancer survivors was a person from Louisville who had been diagnosed with colorectal cancer at the age of 45. And she actually gave a shout out to the Colon Cancer Prevention Project, which is, an effort to prevent colon cancer within Kentucky. So anyways, I think more and more people are realizing that this is a big public health problem, this rise in early onset colorectal cancer, that we do not understand truly the risk factors. We might think of, well, I'm about to say it. So some of the modifiable colorectal cancer risk factors are being obese or having a high body mass index, being physically inactive, smoking, and then dietary factors like having a Western diet or diets high in red and processed meats and high alcohol consumption. So if you think about the rise in early onset colorectal cancer, a lot of the conversation has been because of the rise in childhood obesity, poor diet in the United States, and smoking and physical inactivity. The literature on population attributable risk suggests that around half of colorectal cancers in the United States can be prevented through these modifiable risk factors. So just so I teach epi methods, so I wanted to throw in one of these figures that I teach my students. But if you imagine that this figure is all the total population, and the gray population are those people that are not getting colorectal cancer, we would expect in this total population that there's some baseline risk of colorectal cancer that would just happen even in a perfect world. This light shade of pink is people who are exposed, but whose cancers are probably because of that baseline risk. Now, this orange piece is what we would attribute to colorectal cancer cases who are exposed to these factors. So when I say that half of the colorectal cancer cases are attributed to these colorectal cancer risk factors, we're comparing this piece of the pie relative to all of these cases. So that's the population attributable risk. How does Kentucky compare? Because we know that generally Kentucky is high up there on smoking and physical activity and poor diet. So here I'm presenting again from the American Cancer Society where Kentucky falls on colorectal cancer incidence. Here on the left, we have our incidence rates. These are all of our states. The gray bar is the national average, and you can see, according to the American Cancer Society, we have the second highest incidence rates of colorectal cancer. If you were to go to the CDC website, we'd actually be number one. And so here's our, rates, age standardized rates, around 43 cases per 100,000 people, relative to the national average of around 35 cases per 100,000 people. Kentucky also has one of the highest colorectal cancer mortality rates. Here we are in this dark red, and we're the third highest, have the third highest rates of colorectal cancer mortality, and we are higher than the national average, which is around 13 colorectal cancer deaths per 100,000 people. So we are actually, due to efforts since the early 2000s in our state, we actually have really high colorectal cancer screening prevalence. So here is the prevalence of being up to date for colorectal cancer screening. Now it's 45 years and older. And here we are, and we're in this, gosh, I can't really tell with my eyes, but we're in the top, one of the top states for colorectal cancer screening. So it really begs the question, what's going on in Kentucky? We have high screening, but why do we still have the high incidence rates and mortality rates? If you go further to look within Kentucky, there is quite a lot of variability in colorectal cancer burden in Kentucky. So this data you can get from the Kentucky Cancer Registry, looking at county level rates of colorectal cancer incidence and mortality. You can see that it's really geographically distributed when we look at males. The high counties with high colorectal cancer incidence are distributed across the state. For colorectal cancer in females, it's also distributed across the state, maybe more heavily in eastern Kentucky and the Appalachian region. But there's a lot more variability within a county. Forgot I did this. So if we look at the demographic, racial and ethnicity and gender breakdown within Kentucky, we see again that non-Hispanic Black men have the highest colorectal cancer incidence rate per 100,000 people, and colorectal cancer mortality rate, followed by white men, and then followed by Black women, white women, and then Hispanic men and women. So what we're seeing nationally is also being reflected here within Kentucky. These highest rates within Black men and so on. So we were asking, can we get more of a granular perspective on what colorectal cancer incidence is like within Kentucky census tracts? So I'm going to talk to you about the work that we did to identify colorectal cancer hotspots within Kentucky. We utilized the Kentucky Cancer Registry database and obtained all of the Kentucky primary colorectal cancer cases diagnosed within our state or treated within the state between 1995 through 2018. This includes 56,750 colorectal cancer cases in males and females. The Kentucky Cancer Registry in Kentucky is actually very robust, especially relative to other states. They collect really detailed information for all of these cases. We have information on patient demographics, tobacco use, family history of colorectal cancer, marital status, and health insurance. Tobacco use and family history is not often collected across all state tumor registries, so that's pretty unique to us. We have information on whether or not they are alive or dead, and if they died, what they died from. And we have diagnosis data on tumor grade, nodal involvement, stage, and metastasis site. And we do also have treatment. What's also really great about the Kentucky Cancer Registry is we can get their geocoded residential addresses, meaning we know the latitude and longitude for the residents when they were diagnosed with colorectal cancer. And that allowed for this work. So we calculated sex-specific age standardized colorectal cancer rates for cases diagnosed in that time period in our 1,109 Kentucky census tracts. For each census tract, we counted the number of colorectal cancer cases and divided by the total census tract population size in each census tract to come up with those rates. And we standardized to the sex-specific age distribution. All this is to say, I'm going to show you these rates, and they're not going to be, the rates across Kentucky won't be different because of age or sex differences. Then we use the GEDIS or GI star spatial statistic to identify areas of high age standardized colorectal cancer rate. Once we did that spatial statistic to identify hotspots, we then utilized publicly available databases to learn more about those communities. We utilized the US EPA National Air Toxics Assessment from 2014 to learn about, I'll show you all this later, about ambient metals in the air. We also linked to the American Community Survey and CDC Places databases to get information on neighborhood demographics, social, economic, housing, and lifestyle characteristics of these hotspot neighborhoods. So here are the results from the GETIS GI star spatial statistics analysis. These maps were created by Dr. Matt Ruther and our PhD in epidemiology student, Johnny Newton. And here we are showing the hotspots census tracts in red. So here's a map of Kentucky's 1,115 census tracts. And the areas in black are census tracts that have less than a person. They tend to be the airports and military bases in Kentucky. And the census tracts in red are designated colorectal cancer hotspots. So here's across the state. We're working on publishing this. But for the focus of this talk, I'm going to focus within Louisville. And I've got a map in the next one that kind of blows it up. So here on the left, we have our colorectal cancer hotspot. So here's the airport in black. And you can see we've got this north to south gradient on the western side of Jefferson County, showing our colorectal cancer hotspot. Because we have so many early onset cases within Jefferson County, we were able to present the map of early onset colorectal cancer hotspots within Jefferson County here in the Portland neighborhood within Louisville. So this has been, our interest, one of my interests is examining environmental exposures to cancer. What's really a great website is the EPA's Cleanups in My Community website that generated this figure showing different areas of concern of environmental exposures. So here on the far left, we have one of our Superfund sites. And all of these orange plus signs are brownfield properties or areas where you're not supposed to develop because of environmental contamination. So you can see a lot of these brownfield sites and other sources of environmental pollution are located within the northwestern region of Jefferson County, which happens to correspond pretty closely with what we're seeing in at least northwestern Jefferson County for colorectal cancer. We then decided to focus on metal carcinogens, wondering if these metal carcinogens might be playing a role in colorectal cancer. So just as a backup, there is some literature, epidemiologic literature on metal carcinogens in colorectal cancer, but it's incredibly limited. Ecological studies of arsenic and cadmium, metal carcinogens are arsenic, cadmium, chromium, and nickel. There have been no studies, epidemiologic studies on nickel and chromium, so we focused here on arsenic and cadmium. And ecological studies of arsenic and cadmium have shown positive correlations with having higher age-standardized colorectal cancer incidence rates. There's been one individual level epidemiologic study within the Strong Heart study cohort based out of southwestern US that was looking at arsenic and cadmium in urine samples and followed them up for colorectal cancer mortality. So not necessarily incidence of colorectal cancer, but rather dying from the disease. So there were pretty few, there were only 32 deaths from colorectal cancer in that study. They found some suggestive positive associations with urinary arsenic and colorectal cancer mortality, but it was inconclusive due to the small sample number of events of colorectal cancer mortality. Otherwise, all of the literature related to colorectal cancer has been focused in occupationally exposed populations. And in brief, no epidemiologic study has examined individual level metal carcinogens and colorectal cancer incidence in non-occupational settings. So how does that relate to Louisville? Well, here we're showing maps of arsenic and cadmium in the air from, again, that US EPA NADA database from 2014. So do we have environmental correlates of these colorectal cancer hotspots within Jefferson County? Of all of those Kentucky census tract hotspots, there were 115 of them. 59 of them are within Louisville. So we asked among the 10,243 colorectal cancers that were diagnosed within Jefferson County, we wanted to learn about their environmental exposures to ambient arsenic and ambient cadmium. And so what we observed is that with a 1 standard deviation increase in ambient cadmium levels, these cases had 2.4 times higher odds of residing in one of these neighborhoods. Additionally, the colorectal cancer cases that had a 1 standard deviation increase in ambient arsenic had 2.5 times higher odds of residing in one of these colorectal cancer hotspots. And this was independent of their age, sex, race, family history of colorectal cancer, tobacco use, marital status, health insurance, and neighborhood characteristics like population density, SCS, and screening and physical inactivity, suggesting that perhaps the metal carcinogens in the air might be contributing to living in these cancer hotspots. Okay, so that was a long introduction to what's been the most exciting part, a lot of excitement in this last year. So now we're going to introduce the LEAPS in Colorectal Cancer Study. I really love this acronym too. I was working on breast cancer with a couple of med students, Karen Udo and Alexa Hauser, and we came up with this acronym LEAPS in Colorectal Cancer, which just made me very happy to think of that we might be contributing to, a leap in colorectal cancer understanding. But anyways, side story. Also, I forgot to mention, very importantly, we did this work in 2021, but Dr. Kavalukas and Ali Jen, the medical student at Louisville, were also simultaneously noticing a large number of colorectal cancer cases coming from particular neighborhoods in West Louisville. So we were, 2 ships sailing in the night, didn't know that we were observing similar things in different populations. And Chris States introduced us over email, and we started, Dr. Kavalukas and I started collaborating in April of 2023. So all of this work that I'm about to present has been only within a year's time. So the LEAPS in Louisville Environmental Assessment's pilot study of colorectal cancer is a clinical case control study based out of U of L Health in Dr. Kavalukas' colorectal surgery clinic. Participants are consented and complete a detailed self-reported questionnaire on their current residence and residential histories since 2000 for every 5, so from 2000, 2005, 2010, 15, 20, what we are asking them. We get demographic information, their age, gender, race, and ethnicity. We get information about their housing characteristics, colon health history, and socioeconomic factors like marital status, living alone, income, education, and employment. And we also ask them about their height and weight to derive BMI. We ask about current and past smoking, their physical activity levels, and dietary information on some of those colorectal cancer risk factors, alcohol, meats, and then we added in shellfish and rice because those are contributors to high levels of arsenic. We also ask about frequency of fast food consumption, how frequently they get takeout food and go to casual dining restaurants. And all of these data are entered into REDCap. Right now, this has been an effort. I'll talk about this in here in a bit. But so here's just an example of our questionnaire that we're that the participants are completing. It's an 8-page questionnaire. I'm just showing you all the first couple pages. And for those that are interested in epidemiology, I'll tell you a couple of woes, errors that I committed that could have been easily prevented. So first off, anytime you're sharing documents that you're going to give to a patient, share it in PDF form, not in Word form. Different versions of Word can make it such that parts of the responses appear on a different page than the question. So that was an issue that we have now worked through, but it did lead to some missing data. So, our question on bowel movement frequency, the question was on, page 7, and then the responses were on page 8. We have about 20% people missing data on that one particular question just because of me not sending a PDF version as opposed to a Word version. So anyway, that's the reality of epidemiology. Okay. So they complete that questionnaire.
we are also collecting blood and urine specimens when they're undergoing surgery. So patients that are getting their screening colonoscopies are our controls, and they're those part of that pre-op procedures they're already collecting blood, so we're just adding one additional 5 cc's of blood to collect. Now we are asking them to provide a urine specimen, and so far, people that are providing blood are generally also providing urine, so it's not too much of a burden to ask for an additional urine sample. For the case, our people who are undergoing surgery for their colorectal cancer are our cases, and these 5 specimens are stored in the CIEHS freezers thanks to the help of Louise. Again, I'll introduce more of the team later. And we are actively, with some CIEHS medium voucher funds, actively developing our protocol to aliquot samples with the clinic, with a collaboration with the clinical trials unit, so that we can have smaller samples of serum and plasma and whole blood to run, so that we don't use the entire sample just for one exposure measurement. So here's the enrollment report within around 4-month time period. Dr. Luke Kovalukas and her research study coordinator, Judy Stevenson, have been fantastic, and we've, within a short amount of time, really knocked this out of the park with consenting 89 participants, of whom 34 are colorectal cancer cases and 55 are controls. Now, our questionnaire completion rate is actually pretty good, with 73% completing a detailed 8-page questionnaire. And this is without any sort of monetary incentives or any follow-up outreach to the participants. Of these people who have completed a questionnaire, 24 are cases and 41 are controls. And around 61% are consented participants that have provided both a questionnaire and a biospecimen. So of those 65 participants that have completed the questionnaire within that 4-month period, here's our demographic breakdown. So the average age in the total population is around 59 years old. And as we would expect, the average age in cases is slightly older than the controls, because we know older age is a risk factor for colorectal cancer. This leaves CRC participants, around 49% of them are men, and among the cases, 67% are men. And this is actually quite striking to me how different the proportions are of men in cases and controls. But again, we do know that men have higher risk of colorectal cancer. Here's our demographic breakdown. Around 83% of this population are white, 14% are Black, and 3% are Asian. We do ask about other racial groups. However, no one has reported anything outside of Asian, Black, and white so far. We only have one person who's reported, one person who's Hispanic so far. 22% have a history of colorectal cancer. And as we expect, 25% of cases have a history of colorectal cancer, which is higher than the percentage among the controls, with only 20% reporting a family history. 67% have a history of polyps, and again, we're seeing that within the cases. The percentage is higher for cases than controls. We have one person reporting a family history of Lynch syndrome, who we'll have to exclude when we get to our case control study. And then the average BMI is in the overweight region, with around an average BMI of 29 kilograms per square meter. And we see that that is the average BMI is higher in cases than controls, again, as we would expect. So I'm presenting this data to show you, within a small period of time, we have been successful in enrolling and having good questionnaire completion rates. I'm also showing this data to show that we're seeing what we would expect with the data. So I just think this is all good proof of concept data to present. For smoking, around 38% are either past or current smokers. If we had broken that down into current smoking, around 10% are reporting being current smokers. Here's some of the SES information for these group of participants. You know, the majority of this cohort are married, and here's our total household income breakdown. This is one variable that also kind of has high missing. I think just people are not wanting to report income. But for the most part, our missing data is around 6% for education and employment and other variables with that throughout the month. It's one of those variables where the responses were on a different page than the other than the question. So we have quite a bit of a distribution of income and education and employment within this cohort. So how do our cases in our LEAP study compare to the cases of all of Jefferson County? So among all of the Jefferson County colorectal cancer cases, that's those 10,000 or so, you know, the average age is 68 years old. For our 24 colorectal cancer cases, the average age is 60. So is this because we're seeing the rise in early onset colorectal cancer? Might be that, but also just might be much younger because we still only have just 24 colorectal cancer cases enrolled that have a questionnaire. So the cases in our LEAP study also tend to be more male. We have a much higher 96% of our cases are white, whereas 82% of all of Jefferson County cases are white, and much lower percentage of Black cases than all of Jefferson County. So again, I think this will improve once we keep our enrollment, and we'll probably get closer to that of Jefferson County. But I do think we need to think about future efforts to enroll other racial groups and gender groups to be more in line with that of Jefferson County. And we do have a lot of community partners that I think we could reach out to, but we'll get there. So we received some pilot funding to measure metals in those urine and blood specimens. We are specifically interested in arsenic and cadmium, but we do have metal measurements for an additional around 23 other metals. So we had pilot funds to measure 8 participants' urine and blood samples, and we decided to focus on not current smokers because we didn't want to see variation because smoking is a strong contributor to arsenic and cadmium. So among those 8 participants, 5 were controls and 3 were colorectal cancer cases. 2 were Black, 6 were white, 2 were female, 6 were male, and 4 were less than 50 years old and 4 were older than 50 years old. And 200 microliters of blood and 200 microliters of urine were analyzed to show the metals that I'll present in the next slide. And the main goals were at the time were to look at the correlations between metal levels in the urine and blood. At the time, we didn't have funding, so we needed to figure out, should we go measuring these metals in urine or in blood? But since then, we've gotten some funds so that we can keep measuring metals in both urine and blood. And then we also wanted to do some crude comparisons of the metals between these groups. So I'm presenting here the correlation matrix of arsenic and cadmium. And if we look at the correlation between blood arsenic and urinary arsenic, the Spearman correlation coefficients are high, 0.85. Furthermore, the correlation between blood cadmium and urinary cadmium are also high with the Spearman correlation coefficient of 0.72. Sorry for the poor formatting right there. But it's also interestingly, I would have expected more correlation between arsenic and cadmium, but there's actually no correlation between arsenic and cadmium. If anything, it might be inversely related. So probably there's some basic scientists that know the underlying mechanisms for that, but or have some ideas around that. So here's just some distributions of urinary arsenic, blood arsenic, urinary cadmium, and blood cadmium. So here are the average levels of those metals. You can see that these are quite low in these non-smoking population. Again, this is only 8 people, but here, and we've got our standard deviations here on the far right. And if we look at the distributions in the 3 cases versus the 5 controls, here we're showing the mean of urinary arsenic, blood arsenic, urinary cadmium, and blood cadmium. And on this far right column, we show the difference in those metals between cases and controls. And for urinary arsenic, urinary cadmium, and blood cadmium, levels are on average higher than in the cases than in the controls. The only metal where there was where the cases actually had lower levels of the metal than the controls was blood arsenic. When we conduct our statistical tests, of course, the t-tests are not statistically significant at the 0.05 level, so we wouldn't really expect, given such small sample sizes, that these differences are statistically significant. What I like to look at, though, is how these differences compare to the standard deviation. So I'll just pull up this one because the math is easiest. So for blood cadmium, the difference in blood cadmium in cases and control is 0.0028, and that is essentially 1 standard deviation. So it does look like the colorectal cancer cases have 1 standard deviation increase in blood cadmium compared to the control. Again, not statistically significant, but good pilot data for us to keep going with this analysis. If you want to look at the differences in any of your of all these metals here, it's in alphabetical order. In bold are the arsenic and cadmium, but you can pick out, your favorite metal and look at those differences. The ones that are in gray are where the average level of the metal is higher in the cases than in the controls. And the ones in the blue are where the average levels are higher in the controls than in the cases. But again, t-test p-values were all greater than 0.05, except for calcium. So calcium levels were much higher in the cases than in the controls. Here's blood metal measurements, again, for all of our metals. Same idea, where the values that are in gray means that they had the cases had higher levels in the blood than in the controls, and if it's in blue, the cases had lower levels than the controls. But again, all t-test p-values were greater than 0.05 for all comparisons to the small sample size. So we received funding in this past January to continue this work through June of 2025. We're going to keep continuing enrolling participants in the Louisville colorectal cancer study. We're going to be aliquoting biospecimens, urine aliquots, whole blood aliquots, serum, and plasma aliquots. We'll be able to run those metal assessments for the full population in that time period. Another feature of this grant is we're going to do a validation study of the ambient air estimates of metals to that of the individual level metal measurements in the blood and urine. Also, through this work, we have funding now to geocode the addresses of the participants, both at the time they enrolled and their past residential histories. And we have funds to use LexisNexis to verify those residential histories. LexisNexis is a service that aggregates public databases, so things like tax records and housing records, so things that are publicly available. It's actually quite creepy, but this software allows us to verify residential history. So we will never share our participants' residential addresses to LexisNexis. The way it works is we go in and search the database and verify them ourselves based off of public record. This method has been used in other state registries to verify residential history. So the Georgia State Tumor Registry uses LexisNexis to verify past residences because, so that's going to be an interesting thing, part of the study. So all of these addresses that are being geocoded will allow us to link to environmental estimates. But what's really exciting is we can use these residential histories to construct long-term exposure estimates. So those ambient estimates of the metals that I was presenting the maps up for, we can do that all the way back to 1995. So we can construct what we would estimate their exposure is based off of their residential histories. It'll be the first of its kind to look at residential histories in colorectal cancer. So I'm really excited about this work that Sammy and or Dr. Kavalukas and I received earlier just a few weeks ago. So this has really been a huge team effort. When I was getting my degree in epidemiology, I never would have expected that really part of my job is building teams in addition to the science. So Dr. Kavalukas has been incredible in all of her efforts to help enroll participants, generate great, cool ideas, and establish more collaboration. Luke High has been his lab has run the metals, and Jason has been running those metals. Judy Stevenson is our research study coordinator, who's also been very great at helping getting all of our biospecimens collected and our participants consented and bringing over the questionnaire data so that we can enter it into REDCap. Susan Thornberry is the one who established our REDCap database and has been so helpful. Luis Salazar Guzman has been helping us with getting all of our biospecimens in the freezer and ordering our supplies that we now have funds for to be able to do this work. Allie Jin is our medical student who's been helping with the questionnaire development, as well as helping Dr. Kavalukas with understanding where the patient from Dr. Kavalukas are residing. And also this work has been really through the help of our CIEHS leadership, Chris States and Matt Cave. This work was really initiated with Tyler Ellis. I was, after my first baby maternity leave, Dr. Ellis emailed me, hey, do you want to put in an LOI? And we turned around an LOI in a day on my first day back from maternity leave. And so, yeah, just to, to all the moms out there, you can do it. Matt Ruther has been is our demographer and expert map maker. We've been working with Ted Smith and Rochelle Holm to understand the wastewater metal distribution in these colorectal cancer hotspots and non-hotspots. I forgot to ask them if I could share the data, so I didn't present that today. And then we also have all these other collaborators who have been pivotal in some aspect of this work. So really want to thank everyone for this huge effort across U of L. And also the research support this has really been born out of the support from CIEHS through that pilot award, 2 pilot awards now, and a medium research voucher. So without that, I don't think the study would actually be kicking as fast as it has. So thank you so much for your time and attention. I'm happy to answer questions, and I could probably talk all day to you about other things related to this project, but thanks again, and yeah, happy to answer questions.
Yeah, so great jobs. I absolutely love this project, and every time I see it, I get excited and think of new ideas. So I got to get you in touch with Kate Hepsa. Her name is at UK. So she leads their super fun project. And Kate. Yeah. So she's doing PFAS in colon cancer.
Oh, okay.
But they're doing it primarily in mouse. She's working with Mark Evers. So same type of deal, working with a surgical oncologist. They may be getting patient samples too, but you can very easily make it a multi-site center or study if they were, and just pull resources. You look at the metals, they look at the PFAS.
Do they do the PFAS at UK? Okay.
Yeah, they're super fun. They're kind of quadruple that when they're not measuring horse doping steroids, they measure PFAS. So that's number one. They have animal models too. So Kate has animal models for colon cancer where they're giving PFAS, and we can do those models here too. So we can make a nice translational brand out of this instead of getting PFAS, getting metals. Yeah. And then the second thought I had is, this next specific game would be so you're the CRC lead, so it'll be the CAP lead. So the colon adenomatous polyps, so the colon polyps are the precursor lesion to cancers. You don't actually have to study colon cancer. You can study colon polyps, and so you can get everybody coming in for screening colonoscopy average risk, and some will have polyps and some won't. And so that's your, that's another cohort study. For example, like at our VA, I think we do about 15 screening colons a day, trying to get the numbers for the U of L sites. That's probably at least another 15 or 20. So it's to be very feasible to enroll 25 subjects a day if you're just doing all comers with colonoscopies. And the other neat thing that you can do, which would be really slick, is you could have them take a biopsy of the random colon if there's no polyp, but you could have them take one next to the polyp. If they have a polyp, you can measure metals in that, so you get tissue levels like adjacent to the neoplastic lesion, which might be kind of cool too.
So we just put in, maybe I haven't told you this, but last time we submitted our grant, the R1 grant, in one of the future directions we talked about what do we do after this? And we talked about in the future direction. Can we figure out the relationship between metals and colorectal cancer before the cancer develops? I think, I don't know if we're ready to do it now. I think there's some tissue things going on that.
Yeah, about the amount of tissue metal analysis at 50 milligrams, which is a lot of tissue. Even if we did it on our machines, we were hoping to prove that the having ours and we hired the cancers. But 50 milligrams of a treated rectal cancer is never experience. We're trying to figure out what step in 5 minutes before we try to find out the difference, or if anybody here knows of maybe a way to measure others, it'll be less than 50 milligrams. We can 50 milligrams is still up.
I think this should be able to do less than that. Because we can have them get ICP-MS or adding metals just off a little needle biopsy, which is certainly a lot smaller than the colon.
Yeah, I don't know. I'll have to talk to the methodology, but when we asked him about it, I was like, maybe we would have less.
Okay. We need to turn that M into a micro.
Yeah.
I would bet between all the sites, and that's not even counting the colorectal surgeons, how many you guys are doing, the people coming in for colonoscopies. I bet you can enroll easily 50 a week, probably more than that. You're only being limited by your research staff hours.
Well, I think that's what we're developing right now is just this protocol for how this is happening. And so I think once we kind of get established, I think there is quite a bit of excitement. So expanding would be fantastic. Just need to make sure we can handle it. Yes.
I'm wondering if you were going to be looking at some examples. I don't know if that's the answer, but it's a sample from the environment that you're.
Yeah. So I'm probably going to defer to you again, Andy. On
yeah, I have a study going on these microenvironments samples, and it is it's very difficult. It's a lot more difficult than you would think you get. You don't want to get out of outbreak. You can't get it a colonoscopy. It's going down the microbiome like outbreak thing. I'm able to get it like from the OR. If I do a case, I'll get a case, but it's still like terrified to get exactly. Like a lot of people come to clinic, they don't have to go when they come to clinic. We send them home with a collection canister, but then that relies on them coming back to take it back to you. It's a huge ordeal. We ran into more problems than we, just from a collection standpoint, that we had something for it.
And I think there's something there with the wastewater that just hasn't been maybe fully. It's hard to convince people about these wastewater samples, but I think there's something there with the wastewater samples where we know we can measure levels. And there are groups that can do genomic, or well, we know we can do genomics in wastewater because of SARS-CoV-2. So I think there's something there, but it's wastewater-based epidemiology is still pretty novel. So there might be some we might be able to understand community level metals and colorectal cancer genomics that could be related to why we're seeing neighborhoods with colorectal cancer. So if there's something there that's brewing, we'll see how in our one what the NIH thinks about that idea of wastewater as a marker for environmental exposures that are related to community level rates. I think that's what the uphill battle that we have to try to convince the NIH that understanding community level exposures is relevant for public health. So that's our battle we are currently in. Hopefully.
So if I understand correctly, your basic hypothesis is wherever there is higher ambient arsenic, people are exposed and are likely to be hotspots where we see higher incidence.
So I presented kind of multiple hypotheses. So in this analysis, that is one of them. That the reasons for this hypothesis is among cases, are you more likely to reside in these areas if you have higher levels? And that's what this analysis is showing. What we're doing in the LEAP study is actually more of the scientifically sound individual level measurement. Our individual level environmental exposures associated with colorectal cancer.
So given that the urine and blood concentration is pretty low, is there a way that you can go out there is very low ambient level and show that this exposure in the blood actually higher? Because otherwise.
And that's exactly what the validation study that we're doing will get at. So this study, our participants are generally from Jefferson County right now. And there's some from other regions of Kentucky, some from Indiana, but we'll be able to do that. We can get these ambient estimates and have their individual metal measured concentrations to figure out is what's happening in the environment reflecting what we're seeing in urine and blood. So I think that's going to be a huge piece because these EPA estimates have been used in cancer epidemiology research. And when we think about designing a cancer epi study, the ideal thing would have been to enroll people 50 years ago, measured their metals 50 years ago, so that we would already know by now whether metals are related to colorectal cancer. I don't think we have another 50 years to start that cohort study, which is why a case control study is much more efficient, sampling. And I think if we can answer this validation piece, we'll have some really compelling data that what other epi studies have been doing using the existing estimates might actually be reflecting individual inhalation.
So I think there's going to be just like a base metal sample reading of central water locations in the communities, especially if they're more industrialized districts, because just byproducts like chromium is a large factor industrial processes is often found in water.
to see if those limits are above the EPA standards, and then see if they cause something chromium induced chromosomal instability. That plays a role. Yeah, so I don't have chromium. Hopefully, well, okay. So the wastewater shed are like looking at a map of wastewater sheds, you're going to be like, what the heck? This is really complicated. But you know what we wrote in our grant to kind of tease out what's coming from an industrial source versus what's coming from individual or community waste is you can, if there is a neighborhood where there is an industrial source, you can go above the water line and below. And so above the industrial contamination is going to not be because of the industrial output. So that's one of our proposed methods in our wastewater grant is to kind of get at that. But we, to talk about water samples, yeah. I would love to collect water samples, soil samples. It's just about feasibility now, and studies really kind of kicking off. But yeah, there's a lot of really cool implications for that, and that's a really good point. But I think it's really important. Like, what do we do when we get an estimate of industrial emissions are in the water? So now, now that estimate is about environmental contamination, which I think is the one really important question that we want to know, and not necessarily just throw it out because it's an industrial source. It's just a different type of way of thinking about the exposure. Do we really care about exactly from what an individual with inside an individual, or do we care about the environment around them? Both really important questions that both need to be addressed. Dr. Kim?
I think in my mind that we see a connection between having having a hard time making the between having that colorectal cancer. Do you think that so I did some of the vaccines that need to be there. Just maybe there's some heavy metals.
Yeah, something like that. And so I would have presented the epi literature on these metals in relation to colorectal cancer. And like you kind of were alluding to, there's not much out there on how, in human studies, how metals influence colorectal cancer. What I didn't present is all the biological data that we have on how arsenic and cadmium do have genotoxic effects within colon cells. I didn't put that up here. And also, they cause DNA damage or DNA adducts that are relevant within colon and colorectal cancer cells. So there is some biological data from cell lines and animal studies. I'm sorry to put that up there. But again, those exposures are, tend to be like these high levels that are from the occupational settings. And that is, but we do know that, the real question is do low dose chronic exposures that are in the general population, are those influencing colorectal cancer in the general population instead of occupational settings? So I think that's where our niche is we're going outside of occupational settings, looking at low dose exposures chronically that are influencing colorectal cancer. And then to your second question, just about different sources of metals. Yeah, we were using ambient metals because that's what's publicly available to us when we were doing this. This was all like hypothesis generation. But arsenic, is the people who get arsenic poisoning are getting it because of using, ground well water. And within Jefferson County, we don't have that type of exposure. But, most people are getting their water from, tap water. So that's actually, yeah. With cadmium, the major source of cadmium is actually smoking. So it was important to consider smoking in all these analyses, and that's why we're looking at smokers for metals. But yeah, so there are other ways to get exposed to arsenic and cadmium. We're probably not getting high exposures because of water, because we all are in Louisville are on the metro water system. But yeah, so there's not really a great question, but again, this area, no epi study has evaluated this, and there is biological cell line data that would suggest that these metals can cause colorectal cancer and tumorigenesis. So I think we're really filling a niche and making a leap in colorectal cancer. Well,
I think that I'll let Dr. States, but I want to hear what you think. One thing that didn't answer your question, that the stuff she's talking about is the particular cadmium that's shown to increase colorectal carcinoma through the data. They cancer pathway across it and kind of spark the pathway of follow to cancer. So there is a little bit more supplements data out there on.
So I got a couple questions. I'll start with the response to Dr. Keith's question about ambient versus water. So ambient metals are actually particulates, and those particulates settle in your nasal passages. You end up swallowing a lot of stuff. It's not an issue. Some cautionary things about the sewer system. So Louisville has two types of sewer systems. Like, be aware of where the line is. There's a combined sewer system in both parts of the city that rainwater and everything else moves into. And there's a sanitary along, which is mostly in the southern district funds. So the sanitary will not get the stuff that's in the streets and in the air that gets rained on. And then the other things, the numbers you put up seem very low, and I'm wondering, did you correct those? Because Jason reports concentrations in the diluted sample he injects. It's not back calculated.
Yeah, I still have to follow up with him about that. Yeah, I still have to follow up with him about that.
It's 40 points.
Okay. Yeah, well, that would help out a lot.
Yeah, the solutions that he did. I asked them to back calculate it for people.
And he might have, I just can't fully recall. Okay. So anyways, I guess this is like, it could only get better than this.
The other question I have for everybody else and the other cancers that are near and dear to your heart. I mean, I was having this conversation with my mom out of St. Mary that sent me a lot of. That's why I kind of noticed that a lot of my patients were coming from this southwestern, western corridor. So he sees this uptick in a lot of cancers that he's not been going through. So if there's something else that you're studying, if that was one of the things that I haven't gotten. And I gave us a very preliminary talk and advice on like something interesting. One of the surgical oncologists was like, well, if you look at the rates of thyroid, sarcoma, and non-GI cancer, things like that. I obviously don't have bandwidth to go down that road, but if you guys have something or whatever you're working on, you can also look into other cancers that may or may not.
So I would suggest bladder cancer with the prostate in particular, and prostate cancer with the cadmium.
Yeah, that would be great. And breast cancer.
Or solid organ cancer. That's that kind of. I think they wanted something that was not a GI cancer that like surveys you're like control that it's not just all the cancers are worse because of
I think cover all that and also affecting hepatomas, the hepatocytic carcinomas, the mineralized and background cirrhosis are really fine for this.
It's kind of piggybacks. States in that you is there all day to control the differences in the concentration of your
Yeah, and that was one comparison that I just haven't had time to make or like, yeah, and I think with our funds to do this and, you know, all of our participants of these, just these 8 is, you know, we can actually run prediction models of what are the predictors for high urinary arsenic levels. So, you know, our dependent variables, the metal, and then our independent variables are the ambient levels and smoking. You know, we can figure out what sort of factors are predicting higher arsenic level in urine or whatever the metal is. So that's one thing that we learned about in R1 to better understand that. Like what are the predictors outside of, and yeah. So we don't have water samples though. So yeah.
I think what I'm needing to do is that they kind of he or she on water day. I see turning down water, so concentration.
Right. I think these are all creatinine normalized already. So I should think for bringing that up. I need to insert that in the methodology. But they are.
I'm gonna need to get the information back. Because I didn't have.
But they're also that show. So,
But these are-
but they're so dependent.
I wish Dr. Green here to talk more about that, but yeah, thank you for bringing that up. I need to tease that out. For the next time.
One thing you haven't mentioned yet is that this is just skin cancer and arsenic all over again. So it's intestinal epithelium, it's skin epithelium. So it's basically the exact same thing, just a different epithelium.
Yes, and
This is from Dr. Richard. Really nice talking. I'm just curious, when you mentioned blood measurements, were those blood or serum?
this was whole blood.
Given, so Dr. We had like 20 emails back and forth. You wanted whole blood because then we can find metals and you want to be able to measure that. So we when we collect our specimens now, we are keeping a whole blood in case needs to like measure the levels that are down and moving forward, we're gonna break it down for serum and plasma for kind of future things, but as of right now, it's preferring and
I have a question about the initial survey. So when you asked about smoking history, do you specify cigarettes or e-cigarettes or vaping?
We don't specify vaping. So it's just smoking cigarettes. And we do ask about cigarettes per day. You know, from the background knowledge I have, you know, vaping is much more common in younger populations. So I, and yeah, so I would be kind of shocked if there was a high proportion in this older population. But you're right, we're just capturing cigarettes. Yeah, and so again, 10% of the population is reporting that they're current smoker. And then an additional 28% are past smokers. But yeah, we're not capturing vaping right now. Thank you all so much. I do have to go teach.